Hi, my name is Cyrus, and today I'll be presenting on the spatial and temporal functional organization of the hippocampus and the macaque monkey brain. Just a bit about me before we begin. I'm a rising junior at the Harper School in San Jose, and I'm interested in resting state fMRI, cognitive science, and biophysics in general. So I'm also an avid participant in the US Physics Olympiad, and I write problems for my school's Physics Invitational. And I'm also involved in future problem solving as the director of research in the International Student Advisory Group. And in my free time, I like playing guitar, playing soccer, hiking, and doing physics. Okay, so let's go over a quick agenda for my talk today. I'll be talking about first, giving an overview of the background and the problem, then discussing some of my methods, results, and finally, the conclusion for my research and some further avenues of exploration. So let's first talk a little about global health and resting state networks. So the brain reacts differently over time to real-world sensory input. And this means that for the past decades, neuroscientists have been looking to understand how the large-scale organization of the brain models the environment. And they've also found that these neuronal networks, that's what they're called, are present even at rest. So when you're not doing any tasks. And it's important to understand these networks because disruptions to them have been associated with many neurological diseases such as Alzheimer's disease, epilepsy, and schizophrenia. So we can see a quick diagram here. You can see the healthy control baseline and the Alzheimer's disease baseline. And as after 1 year, you can see the functional organization, and you can see the hubs represented by those spheres are markedly different in the Alzheimer's disease patients. So my goal is to study and understand the relation between the temporal hierarchy and anatomical organization in the brain. So I also aim to validate the use of non-invasive techniques in the hippocampus, since as of now, the hierarchical organization of the brain is studied primarily via invasive intracranial recordings. Okay, so let's talk a bit about timescales. So let's say, for example, you trip and skin your knee. So how do you respond to this? Well, the short answer is your brain operates across multiple timescales. I'm going to clarify what that means in a second. So in the milliseconds and seconds after you trip, your brain's primarily concerned with sensory input and motor control, and this is often pre-trained and automatic. Then this is all embedded in a broader temporal context, that is in the minutes after, where you're assessing the injury and determining preliminary treatment. And this is all in an even slower time scale, that is the hours after, where you're primarily concerned with recovery and long-term treatment. So your response to tripping here is the result of integrating and processing information over multiple timescales, and when you're trying to achieve the goals that are embedded in these hierarchical timescales. And you can see various fast timescales that are modulated by slow timescales and vice versa. So a logical next question is, how does the brain do this? And the answer is, in short, they require different brain states. So one way of approaching this question of how does the brain model these timescales is by looking at the innate ability of the brain to integrate information over different timescales. So for example, in 2014, Marin colleagues showed that neurons have different timescales at rest, and they revealed a hierarchy extending from sensory to association areas. And in this case, association areas just refers to areas that deal with abstract processes, such as planning. So they demonstrated that unimodal areas that deal with sensory input have fast timescales, and the aforementioned association areas have slower timescales. So the organization of the brain has been described as a map, but it remains to be seen whether distinct regions or continuous gradients are a better representation. But in recent decades, various researchers have published papers, and they, for the large part, shown that these continuous gradients, a gradient-based organization, is a better way to describe intrinsic neural timescales across the brain. So these are called functional connectivity gradients, and they can uncover principles of functional organization, such as, for example, the homunculus and the primary motor cortex. So gradients are establishing themselves as a new powerful way to look at the brain's organization. In my research, I hypothesized that one gradient dimension of the functional connectivity architecture of the brain is closely linked to timescales. And in the past, the relationship between timescales and functional connectivity gradients in macaque monkeys has been established in the cortex and in the striatum, but not yet in the hippocampus, which is what I aim to prove in my project. So let's talk a bit about the hippocampus first. So it's nestled in the temporal lobe, as you can see here, primarily concerned with memory processes, although of course it has various other functions as well. So it also demonstrates what's known as longitudinal access specialization. What this means is that the aforementioned functional connectivity gradients are organized from anterior to posterior in the hippocampus. So you can see in the diagram here, and in one more diagram, that this longitudinal axis specialization is present in the hippocampus, and I'm going to verify that too in my research. So I aim to discover what is the relationship between temporal and spatial dynamics in the hippocampus. So to do that, I utilized data from 4 macaque monkeys who are first, anesthesia was first induced into them, and then their fMRI data was acquired using a 10.5 Tesla magnet that was pre-processed using various correction and smoothing algorithms. And finally, I applied analysis and tried to discover the relationship between intrinsic neural timescales and functional connectivity gradients. So just a bit on how I actually calculated these intrinsic neural timescales. I adapted a technique from Watanabe and his colleagues in 2019, where they used something called the autocorrelation function to calculate these timescales. So as to demonstrate the functional connectivity gradients, I adapted a technique from Vosta Wall and colleagues in 2020, where they essentially correlated voxels in the hippocampus, or they correlated voxels to obtain a functional connectivity matrix. And I did this in the hippocampus. And then they applied what's called a cosine similarity kernel to obtain what's known as the similarity matrix, affinity matrix. And then this matrix basically captures the connectivity fingerprint of each voxel. Finally, I applied diffusion embedding, and then that's how I determined the functional connectivity gradients. And I projected these onto the cortical surface for more analysis. So to evaluate the FC-INT link, which is the functional connectivity intrinsic neural timescale link, I looked in the hippocampus and I discovered that it had a Pearson's correlation of 0.4304 at the group level. So this is a significant correlation, and you can see here the anterior to posterior gradients, as I mentioned before in the hippocampus. At the subject level, we saw a similar correspondence, so anterior to posterior organization again, and we can see a R of 0.4798. So again, correlated meaningfully. So to conclude on what I talked about today, we determined that information is processed across multiple timescales, and that gradients estimated from functional connectivity reveal the functional organization of the brain. And it's important to understand this functional connectivity because disruptions to these networks are often associated with diseases, as I mentioned before, such as Alzheimer's disease and epilepsy. So we determined, again, within area connectivity is meaningfully correlated with intrinsic neural timescales. However, we saw that whole brain connectivity, that is, you correlate the voxels in the hippocampus with the entire brain, is uncorrelated with timescales. And we hypothesized that's because that when you correlate with the whole brain, you have multiple functional connectivity maps overlapping, which disrupts the connectivity. So finally, I'd like to thank the contributors listed on the screen for making this research possible, and thank you for listening. And if you have any questions, I would love to hear from you at my email listed on the screen.